And we'll move on to the final talk of the session. So it's a huge pleasure for me to introduce Simin Meydani from the Jean Mayer USDA Human Nutrition Research Center at Tufts in Boston. And Simin is really the global expert on vitamin E and immunity, and that's the topic of the presentation. So over to you, Simin.
Thank you so much, Philip. And so it's a pleasure to be talking to you today. I'm going to focus my presentation on older population, mainly because they are the fastest growing group of people. By the year 2050, as you can see there, we are going to be having 30% of the population being over the age of 65. They're also most susceptible to infections, particularly respiratory infections, that it has been very well demonstrated by the recent pandemic of COVID-19. Not only older people are more susceptible to respiratory infections, but they have higher morbidity and mortality from them. A very important contributor to this higher susceptibility to infection in older people is the dysregulation of the immune function. As Philip pointed out earlier, we need both the cell-mediated immunity, particularly T-cell mediated function, as well as the inflammatory responses to respond well to viral infections. And if you have hyporeactivity of the cell-mediated immunity, you get more susceptible to infection. And if you have hyperactivity of the inflammatory responses, you get higher morbidity and mortality from them. And in case of older people, it seems to have the perfect storm in that they have lower cell-mediated immunity, particularly of the T-cell mediated function, and higher inflammatory responses, which results in the cytokine storm. So, for example, in many indices or indicators of T-cell mediated function, such as response to vaccines and the ability of the lymphocytes and T-cells to proliferate or to have a cytotoxic reaction to the virus, goes down with aging. For example, the flu vaccine is only 40% effective in older population. And as I mentioned, the ability of the T cells to proliferate, to produce key cytokines, goes down with aging. These are just a couple of examples of this. This is looking at the ability of the cells to, from mice, young and old mice, to proliferate, and you can see there's a significant reduction, as is their ability to produce a key cytokine, for example, IL-2. This is, there is of course different variation in terms of how much decline you would observe, particularly in humans, but nevertheless, it's consistently observed. This is also looking at, this inability to proliferate and produce cytokines is both due to changes intrinsically in the T cells themselves, as well as to production of suppressive factors, such as, for example, prostaglandin E2. Here you're looking at the ability of the T cells to go through, naive T cells, for example, to go through cell cycle division. You can see that the young cells can go through several cell cycle divisions, but in the olds, you can see that they're pretty much stuck in zero, and there's very little that goes through 1 and 2 cell cycle divisions. On the other hand, if you look at, for example, of the ability of the macrophages to produce cytokines such as IL-1, IL-6, and TNF alpha and IL-10, you can see that there's significant increases in the cytokines. And one particular of interest in terms of T cell function is the significant increase in PGE2 production with aging, and also in light of what was discussed earlier in terms of fish oils. And this is observed, of course, in other tissues, not just in the macrophages. This is looking at pro-inflammatory cytokines and expression of pro-inflammatory genes, such as COX-2, IL-1, and TNF-alpha in adipose tissue. And we've done, though I wouldn't have very much time to go through this, but we've done a lot of work to show that this increase in pro-inflammatory genes is to a large extent due to higher production of sphingolipid ceramide in the aged. Now, there has been a lot of attempt to change this age-associated dysregulation of the immune function through dietary interventions, and several things have been attempted. We will focus just on vitamin E given the time and what was requested of my presentation. So vitamin E, as you know, is an antioxidant that is found in plant oils as well as in different nuts, particularly almonds. Several clinical and preclinical studies have shown that vitamin E has antioxidant and anti-inflammatory properties in the aged. I'll show you a couple of examples. This is from the work that we've done in mice, macrophages of mice. You can see that vitamin E supplementation does not decrease, for example, PGE2 production in young macrophages, but it does significantly decrease it in old macrophages who, to begin with, have a higher production of PGE2. In humans also, we have shown that, obviously if you increase the level of vitamin E in humans. This is a study in which we supplemented healthy older people with 800 international units per day of vitamin E. And you can see increase in plasma vitamin E level associated with significant reduction in the ability of peripheral blood mononuclear cells to produce PGE2, and also decreased level of lipid peroxides in the plasma. Similarly, Van Chitts had shown that supplementation with 600 IU per day of vitamin E for 6 weeks in both hyperlipidemic and normolipidemic subjects results in significant reduction in production of pro-inflammatory cytokines such as TNF-alpha, IL-1 beta, and IL-8. On the other hand, we've shown, as well as others, that vitamin E supplementation of healthy elderly can significantly improve in vivo and in vitro indices of cell-mediated function. I would not have time to show you all that data, but here's a couple of slides from first in preclinical studies in mice, and then I'll show you some results in humans. So here you can see that supplementation of old mice with vitamin E can significantly increase ability of the lymphocytes to proliferate. This is showing the change in proliferation after supplementation, not a significant effect in young, but also you can see that there is significant increase in IL-2 production. And if you look at the ability of the cells to go through cell cycle division, again, you can see that this is in the young. You can see old, not so many in the cycle. They can't go through several cycles, but in the animals who are supplemented with vitamin E, old animals, you can see that they can significantly improve their ability to go through more than one cell cycle. So the effect that vitamin E has is both direct on the T cells themselves, and as you can see, but also through inhibition of suppressive factor for T cells such as PGE2. Now, this effect of vitamin E is manifested at the signal transduction level. Here, what you're looking at, one important function in terms of the ability of the T cells to proliferate or produce cytokines is forming these immune synapses at the where there's contact between antigen-presenting cells and the T cells, and you're looking at an effective immune synapse here and an ineffective immune synapse formation here. So in old, there is their ability to produce effective immune synapse goes down, But following vitamin E supplementation, you can see that there is significant improvement in the ability of the T cells to form effective immune synapses. This work was from in vitro supplementation, but you can also see this ability to improve effective immune synapse formation by feeding vitamin E in vivo to old animals as well. So the way that vitamin E can improve T cell function in older adults, we have through a lot of work, we've shown that there are two mechanisms. One is indirectly by reducing PGE2 production, and the other one is by having a direct effect on T cells, particularly of naive T cells, by improving signal transduction. And here's one slide from human studies so that you can see that these results are transferable to humans as well. We did a study in which we supplemented healthy older people with 60, 200, or 800 international units of tocopherol and looked at changes in antibody response, in this case to hepatitis B, and also an in vivo measure of T cell mediated function called DTH. And you can see that following supplementation, we can significantly improve both the response to vaccine and the DTH response. So to summarize what happens in terms of vitamin E and immune and inflammatory responses in the aged, in a way, vitamin E can balance the dysregulation of immune and inflammatory responses in older people. On one hand, it can improve T cell mediated function, and on the other hand, can reduce inflammation. And the two of them are actually, the two functions are related to each other. Now, what happens in terms of vitamin E and infection? Before I get to some of the results that on actual infection, I want to point out that the earlier work by Hannett had shown that following, for example, influenza infection, there is a significant reduction in the level of antioxidant nutrients, particularly of vitamin E and glutathione. There is reduction in vitamin C, but doesn't reach significance. And it was interesting to observe that it seems the same phenomenon happens with a member of coronavirus family, the bovine diarrhea virus. So you can see the change in alpha-tocopherol level following infection in serum. And of course, this was also demonstrated with vitamin D level as well, but not with the other vitamins. So I think this is something to keep in mind, that there seems, and I think there was earlier a question asked about this, what happens with the infection itself to fish oils, but in case of the antioxidant, there does seem to be a significant reduction caused by the infection in the level of the nutrients, so that they might increase the requirement for vitamin E and perhaps vitamin D in case of the infection. So we've done both preclinical studies and clinical studies, as well as others, to look at the effect of vitamin E supplementation on respiratory infection in young and old, young and aged. And here's a study we did by supplementing young and old mice with 500 parts per million per day of vitamin E, or the adequate level of vitamin D, and then exposing the animals to influenza infection. And what you can see here, there wasn't a significant effect as much in the young animals, some effect, but not as dramatic as you can see here. You're looking at long viral titer and days post-infection. You can see that the animals who are given the 500 parts per million of tocopherol have much lower viral titer compared to the ones who are not supplemented. And here is from a longer term study we did in mice again, looking at the effect of vitamin E. Again, you can see that supplementation reduces viral titer, but also what I would like you to note is that it prevents viral induced inflammation. Because after infection, you can see a significant increase in IL-6 as well as TNF-alpha, but animals who are supplemented don't show that significant rise, so prevent that increase in inflammation. And that is reflected in the clinical outcome. As you can see, while the animals who are not supplemented lose about 7 grams of weight and are eating about 6 grams of food. The animals who are given vitamin E don't lose as much weight, and that's because they're eating much more. Their appetite is not being affected, which is related again to the reduction in the inflammatory cytokines. We've also looked at bacterial infection. For example, here is a infection due to pneumococcal step pneumococcus, and you can see that we did an experiment in which young and old mice were given either 30 or 500 parts per million of vitamin E for a month, and then they were infected with S. pneumonia. And what you can see here, again, you can see that the effect is in the older animals. First of all, older animals have higher lung titer of the bacteria compared to young, but those who are supplemented with vitamin E have significantly lower level. The effect is not significant in the young. And this is also reflected in the pathology and in the reduction in the inflammatory cytokines in the lung, as you can see here and here, and the pathology in the lung. This is uninfected, infected, and then you can see that the animals who are not given additional level of vitamin E have a lot more pathology in their lung compared to the ones who are given the vitamin E. And again, these animals don't lose as much weight, the animals who are given the vitamin E, whereas the ones who are given just the recommended level of vitamin E, they do lose much more weight. And then we have done also, this is reproducible in humans. We conducted a study in which we gave nursing home residents 200 IU per day of vitamin E or a placebo for a period of a year. And then we documented their their respiratory infections. We saw a significant reduction in risk for all respiratory infections, but it was mainly reflected in upper respiratory infections, particularly common cold. There was an effect on pneumonia, but it depended on the genotype of the subjects. So what I wanted you to also notice is that in this study, we looked at the different viral and bacterial infections. And as you can see that many of the respiratory infections that we had observed in this study were due to viral infections, including some of the coronavirus types. Of course, these are benign types of coronavirus. At the time, we were not looking for any of the more pathogenic type of coronaviruses, but it might imply that perhaps there could be an impact on some of the more pathogenic type of coronaviruses as well. And recently there was a publication from UK where they looked at the association between vitamin E, D, and A and self-reported respiratory infections, and they did report that there was a negative association between vitamin E and A, and also vitamin D with respiratory infections. The association with vitamin D was more with the supplemental level, and with vitamin E, and A was with the dietary level of the nutrients. So to summarize, at least in the older population, our data as well as others suggest that higher than the currently recommended level of vitamin E would be needed to balance the immune and inflammatory responses in the older adults, because it does decrease in the level of inflammatory cytokines, lipid peroxides, and PGE2 on one hand, and on the other hand, it can improve cell-mediated immune function, particularly of the T-cell mediated function, and that overall would result in reduced infections. We don't have any direct data in COVID-19 patients, but what we have observed in healthy older adults would suggest that perhaps there would be benefit from vitamin E in case of both, I would believe, prevention as well as reduction of the cytokine storm by providing higher levels of vitamin E. And with that, I would like to stop and thank you for your attention.
Great. Thanks so much, Simin. Again, an excellent overview of complex literature, preclinical and clinical. That was really good. There aren't questions that have come in, but I've got one for you, which is, we've heard about omega-3s, we've heard about vitamin D, we've heard about vitamin E. These all seem to be rebalancing the system to favor immunity and dampen inflammation. So it's sort of resetting the balance. But we've talked about each of those nutrients individually. Of course, we don't really consume them, at least in a diet, individually. So how do you see a mix? You know, are there interactions amongst the things we've been talking about, for example?
Well, thank you so much for asking that question. And because I think, as you know, Phillips, at least our earlier work with fish oil in the older people suggests that it would, while you can reduce inflammation in older people with fish oil, but it could have some unexpected effect in terms of T-cell mediated function. And then we've done studies to show that actually if you use a combination of vitamin E and fish oil, you would see the beneficial effect in terms of reducing inflammation, but also keeping the effect, but not having any unwanted effect on T-cell mediated function. So I would think that actually, at least in case of fish oil and vitamin E, a combination will be very beneficial rather than using fish oil alone, but at least in the older population where they do have higher sort of need for antioxidants because their enzymatic antioxidant level goes down with aging and they have higher sort of lipid peroxidation. So I think it will be very important to keep these two things in mind in terms of combining and I mean, the nutrients don't work in isolation as you know, so I think it would be important to keep that in mind.
Yeah. I think another thing, maybe this will be the last question because we're sort of almost to time. Another thing which has come out, particularly in your talk, and it's relevant to COVID-19, is the different impact of immunity and inflammation, but also that nutrients can have according to age of people. And I wonder whether you think you know, there are particular groups of the population who really need these nutrients more than other groups.
I do. I think particularly if we're just talking about healthy people, I do believe that for at least our work indicates that older people would need higher level of vitamin E, but that's not necessarily true for a healthy young person. Now that might change if there is other types of stressors like infections or environmental stressors or obesity or what have you. But I do think that there is interaction between age and the nutrient requirement, something to keep in mind as well.
Okay, great. So Simin, I think we need to wrap up now. Thanks to the other 5 speakers. I think this was a really excellent session. And it was good to delve away from the fatty acids into the fat-soluble vitamins. I think they're really important. And I think we really combined the existing knowledge with some of the state-of-the-art real-time findings around COVID. So this has been a great session. Thanks to the speakers. Thanks to Wiley's Finest who supported us. And I hope the delegates have learned something new from the session. And I'll just say goodbye to everyone and thanks again.
Bye.
Thank you. Bye.